I was just reading the latest clinical data on inflammatory signals in the aging brain, and it changes how we should evaluate neuroinflammation levels. The aging brain exhibits persistent immune activation. We often assume that age-related cognitive decline is simply a general wear and tear of neural connections. However, the biological reality points to a specific chronic inflammatory signal coming from within. By looking at the cellular level, we can track exactly how this signal spreads. This shifts our understanding from vague cognitive symptoms to a precise mechanism of cell-to-cell communication network breakdown. The primary sources of this persistent signal are senescent cells, which have ceased dividing but remain highly active. They undergo a profound change in their profile, secreting numerous dangerous inflammatory molecules. Under healthy conditions, senescent cells are rapidly cleared by the immune system to allow tissue repair. In the aging brain, however, this clearance mechanism slows down, allowing these cells to persist. Their persistence creates a chronic inflammatory microenvironment that slowly spreads through surrounding tissues. Before neurons show structural damage, they are exposed to this local, continuous inflammatory pressure that impairs their function. As these senescent cells accumulate, they begin to compromise cognitive reserve, which is the brain's resilience to damage. The observable clinical result is a gradual decline in processing speed and memory. Senescent astrocytes and microglia are particularly problematic because they directly support neuronal health. When they become senescent, they stop performing their critical maintenance functions, leaving neighboring neurons vulnerable to stress. Simultaneously, they actively secrete pro-inflammatory cytokines, chemokines, and matrix metalloproteinases. This specific secretion profile, known as the senescence-associated secretory phenotype, directly disrupts the delicate chemical balance required for healthy neural signaling. Separate the mechanism from the outcome. The visible loss of cognitive performance is not just random degeneration, but the direct consequence of this persistent cellular signal undermining synaptic connection function. This inflammatory environment impairs synaptic plasticity, which is the brain's ability to strengthen connections. As a result, the neural networks struggle to encode new information or retrieve older stored memories efficiently. This creates a metabolic and functional bottleneck. Strained neurons must work harder to transmit signals through an inflamed environment, accelerating their own cellular stress and driving further decline in network efficiency. To understand this process, we must define cellular senescence. It is a state of permanent growth arrest triggered by cellular stress, DNA damage, or telomere shortening in active brain cells. This arrest is a protective mechanism that prevents damaged cells from replicating. However, in non-dividing cells like neurons, a similar senescent-like state can occur, characterized by altered genomic gene expression. Now, this altered state changes how these cells interact with their neighbors. Instead of supporting synaptic function, they begin to release signals that actively degrade the surrounding local extracellular matrix. This extracellular degradation disrupts the physical structure that supports synaptic connections. Without a stable matrix, the receptors on neurons cannot align properly, which directly impairs the transmission of crucial electrical signals. Under normal conditions, transient senescence actually helps tissue remodeling and development. But when it becomes chronic, the continuous exposure to these inflammatory signals alters the behavior of healthy neighboring brain cells. For instance, healthy microglia are the resident immune cells of the brain, constantly clearing cellular debris. But when exposed to senescent signals, they adopt a dysfunctional state, losing their phagocytic capacity. This dysfunction leads to the accumulation of damaged proteins and metabolic waste in the brain tissue. The brain's self-cleaning mechanism is compromised, further exacerbating the local inflammatory environment around local neurons. This accumulation of cellular waste creates a feedback loop. The waste itself triggers further cellular stress, pushing more neighboring astrocytes and microglia into a senescent state, spreading the localized inflammatory signal. The core mechanism begins when DNA damage triggers the activation of specific cellular pathways. This activation leads to the persistent production of inflammatory cytokines like interleukin-6 and tumor necrosis factor molecules. These cytokines bind to receptors on healthy neighboring neurons, activating intracellular signaling cascades. This activation leads to the phosphorylation of key proteins that regulate local synaptic transmission and receptor density. Consequently, the density of glutamate receptors on the postsynaptic membrane decreases. With fewer receptors available, the neuron requires stronger stimulation to fire, raising the threshold for successful cell-signaling communications. Simultaneously, the inflammatory environment drives the activation of enzymes that degrade synaptic proteins. Critical components like synaptophysin are depleted, reducing the neuron's ability to release neurotransmitters into the local synaptic cleft. Follow the evidence boundary. We are analyzing a pathway where chronic inflammation alters the physical structure of synapses, leading to silent networks long before any structural cell loss actually occurs. This structural alteration is accompanied by a decline in mitochondrial function within the neuron. The inflammatory signals impair the mitochondrial electron transport chain, reducing the production of adenosine triphosphate energy molecules. With less energy available, the neuron struggles to maintain the ion gradients necessary for electrical signaling. The sodium-potassium pumps slow down, causing the membrane potential to fluctuate and signaling to fail. This energetic crisis is compounded by a decrease in brain-derived neurotrophic factor. The expression of this key growth factor is suppressed by chronic inflammatory signals, halting synaptic repair and local neuroplasticity. Without neurotrophic support, the dendrites of neurons begin to retract. The branching structures that receive signals shrink, reducing the total surface area available for synaptic connections in the neural network. So the network loses its redundant pathways, which are essential for maintaining cognitive reserve. The brain can no longer bypass damaged areas, making cognitive performance highly vulnerable to any further stress. This vulnerability is particularly evident in high-demand cognitive tasks. The brain must recruit additional, less efficient regions to perform simple operations, consuming more energy and causing rapid mental signaling fatigue. At the same time, the blood-brain barrier is compromised by the inflammatory signals. Matrix metalloproteinases degrade the tight junctions between endothelial cells, allowing systemic inflammatory molecules to enter the active brain. This influx of systemic molecules further activates the resident immune cells, reinforcing the local inflammatory state. The brain is now exposed to both local senescent signals and systemic inflammatory stress factors. This dual pressure accelerates the rate of cellular senescence in the cortex. The number of senescent cells rises, creating widespread patches of tissue that can no longer support healthy neural activity. This systemic spread marks the transition from localized cellular stress to a widespread loss of cognitive reserve. The brain's capacity to adapt to challenge is systematically eroded by this chronic signal. To evaluate this model, we must establish its boundaries. Not all brain regions are equally susceptible to cellular senescence, as some areas maintain high clearance rates even in very advanced age. For instance, the cerebellum shows a lower accumulation of senescent cells compared to the cerebral cortex. This regional difference suggests that local microenvironments play a critical role in local cellular aging. Additionally, this mechanism is primarily mapped in preclinical models. Directly measuring the secretory profile of individual senescent cells in a living human brain remains a major technical challenge in contemporary research. The important limitation is we must distinguish association from causation. While senescent cells accumulate in aging brains, they coexist with other aging hallmarks like proteostatic collapse and structural mitochondrial dysfunction. We must also note that cellular senescence is not entirely negative. It remains an essential barrier against oncogenesis, preventing cells with severely damaged DNA from replicating and forming dangerous tumors. However, in conceptual models where senescent cells are selectively cleared, researchers observe a preservation of cognitive function. This provides a validating case for the causal role of these cellular inflammatory signals. By defining these boundaries, we clarify our focus. We are addressing a specific, cell-mediated inflammatory process that contributes to cognitive decline rather than a universal or inevitable biological aging program. This brings us back to the primary question of why inflammation rises in the aging brain. The answer lies in the accumulation of senescent brain cells and the decline of clearance. The persistent secretory profile of these cells creates a self-sustaining cycle of inflammation. They recruit immune cells that, due to age, fail to clear them, leading to chronic neural tissue stress. By understanding this pathway, we can identify specific targets for intervention. Rather than attempting to suppress all inflammatory signals, we must target the specific cells generating this chronic secretory phenotype profile. This strategy focuses on clearing senescent cells or modulating their secretory profile. In laboratory models, these approaches reduce the local inflammatory burden and restore synaptic plasticity in the local hippocampus. Restoring synaptic plasticity allows neurons to rebuild lost connections. The brain can re-establish the redundant networks that support cognitive reserve, improving its resilience to future metabolic or local inflammatory challenges. Simultaneously, reducing the secretory phenotype protects the blood-brain barrier. Tight junctions can be restored, preventing the entry of systemic inflammatory factors that exacerbate the local immune response in local brain tissue. With the inflammatory pressure removed, neighboring astrocytes can return to their support roles. They resume clearing glutamate from the synaptic cleft, preventing excitotoxicity and maintaining a clean signaling environment. Health span over lifespan. The goal is not to artificially extend the life of damaged cells, but to preserve the functional integrity of the neural networks that support overall cognitive performance. By targeting the source of the inflammatory signal, we address a primary driver of cognitive decline. This shifts our clinical perspective from managing symptoms to protecting the brain's local cellular environment. In practice, we can monitor markers of systemic inflammation that may reflect the brain's environment. While direct measurement is difficult, markers like C-reactive protein provide a useful systemic context. Supporting cellular clearance mechanisms involves optimizing our metabolic health. Regular physical exercise supports the immune system's capacity to identify and clear damaged and senescent cells in our peripheral tissues. Another practical consideration is dietary composition. Consuming foods rich in natural polyphenols may support cellular stress resistance, helping cells avoid entering the senescent state under normal human physiological conditions. What matters clinically is, these behavioral approaches do not replace medical therapies. They represent low-risk strategies designed to support baseline cellular health and promote our general cognitive resilience. Optimizing sleep is also critical. During deep sleep, the glymphatic system clears metabolic waste from the brain, potentially reducing the cellular stress that triggers this cellular senescence pathway. Additionally, maintaining stable blood glucose levels minimizes advanced glycation end products. These compounds can bind to receptors on microglial cells, triggering inflammatory pathways that mimic chronic senescent signaling. Measure what is actually supported. When designing a protocol, focus on habits that have clear mechanistic backing for reducing systemic inflammation, rather than chasing unverified and speculative cellular longevity claims. This educational analysis is not individualized medical advice. Consult a healthcare professional before making major changes, especially if you are managing chronic inflammatory conditions or outstanding cognitive concerns. We must evaluate neuroinflammation by targeting the cellular sources that erode our cognitive reserve. Optimize the protocol, protect your brain, and I'll see you in the next data set.